Hello everyone, I am Biotechnology, working as an assistant professor in GTHU College Department of Biotechnology. Today we are going to start with your practical course in food biotechnology under Stride scheme. And today we are going to discuss practical number 3, that is estimation of protein, carbohydrate and chlorophyll from the single cell protein, that is spirulina, cholera and yeast. Before going towards our topic, we have discussed some part of this practical in earlier lecture. So you have discussed estimation of carbohydrate and estimation of chlorophyll. Now, in this session, we are going to study estimation of proteins from yeast cells. This session agenda of our this practical is to learn about the yeast, which is a single cell protein, then the protein content in the yeast. There are various different yeast products which we consume in our day-to-day life, and these yeast are also used for baking or for making some other food products. That protein content we are going to study today. And last is the estimation of protein from that yeast. Yeast as a microorganism. You all know what actually yeast is, right? So in this session we are going to study this microorganism and we are going to list out some products which are actually being produced by yeast. Yeast is a microorganism made up of just a single cell. It is a unicellular microorganism. Means what? A single cell is responsible for doing all the functions of that cell. Means from cell division to the reproduction. Okay, so everything can be done by this single cell microorganism. They can grow through a process called as budding. Budding is an asexual reproduction process which is followed by the yeast cell. So a small yeast cell grows on the outside of a mature one until it is fully grown and ready to separate. So budding is a process where a single cell microorganism produces a different cell, it divides itself and it produces a different cell, and unless and until it is fully mature, it doesn't get separated. So once the cell is fully mature, the one cell gets divided into two completely different cells. They need sufficient food, that is sugar, and agreeable conditions. What are agreeable conditions? Like a proper pH, proper temperature, amount of liquid, amount of sugar, that everything is needed in a proper condition so that the yeast can be grown. So yeast is the most easiest microorganism which can grow very easily, and it takes small period of time for growing. Like for baking bread, okay, so you may have baked bread at your home, so at that time you have used yeast, and that yeast is kept aside for half an hour or 1 hour to raise or to cool, right? So that is a type of yeast, and this yeast is grown very easily at any temperature and at any suitable conditions. Wine, beer, and a lot of breads wouldn't exist without this yeast. So in this figure, you can see the budding process carried on by the yeast. And there are many food products like wine, beer, or breads, which is not possible to make without yeast. Now you can see that we use yeast to make a lot of different foods. So yeast can convert sugar into alcohol through a process called as fermentation. You have studied what fermentation is, right? So production of alcohol, right? Yeast is a microorganism which is actually converting your sugar into alcohol, which is used in making wine and beer. Yeast can also produce carbon dioxide. So that is the reason why your bread gets risen up, right? So because of carbon dioxide production only, the bread is more fluffy, is more soft. It ensures that your bread rises while cooking. Okay, so production of carbon dioxide by the yeast is the reason of rising your bread. In this figure, you can see small air gaps, right? That is nothing but the production of carbon dioxide by the yeast in that dough. Baker's yeast. Okay, so you may have heard some different types of yeast which we use for making bread, for making beer or alcohol. There are different kinds of yeast which can be used for different purposes. So baker's yeast is a species of yeast known as Saccharomyces cerevisiae. Okay, this is a type of yeast that used to bake a bread or to brew a beer. Okay, so beer production, or you can say bread production, is much more easier when we use baker's yeast. There are other alternatives for making bread, like you can use baking soda or baking powder for making your bread, but the benefits of using yeast is much more greater. Like your bread will not raise higher if you're using such type of soda or baking powders. Okay, so whereas if we use yeast, it will raise higher, it will be more softer, and the taste of the bread will also change. While brewers and bakers and nutritional yeast are technically made from a same species of yeast, they are very different products. Okay, so brewers means for brewing beer, for bakers means you have to bake bread, and nutritional. So nutritional is a type of yeast which is generally consumed as it is, or it can be added in various food products. Okay, so it's a directly eatable yeast. Okay, but this type of yeast is not used in India. So in foreign countries, many people consume it because it has high amount of vitamins, minerals, and proteins in it. Types of yeast. There are three major types of yeast, as we have discussed in the earlier slide. You can see that baker's yeast, brewer's yeast, and nutritional yeast. Right? So in baker's yeast, the word itself says that for baking. Right? Baker's yeast means what you have to use that yeast for baking bread. Right? So the baker's yeast is purchased alive and used to bake a bread. So it is purchased alive means what? You may have seen or observed yeast under microscope. Okay, so you may have seen that they are motile, right? You can see moving, or you can see such minute organisms under that microorganism. But when you buy that yeast in market, okay, from any grocery shop, you can see some type of active dried yeast written on that packet. So what does that active means? It means that it is alive. Okay, so alive means that type of alive where you can directly use that product or directly use that powder and allow it to grow in a form of sugar solution, okay, or milk which has sugar, because yeast needs sugar for growing, right, for multiplying. So that type of alive where you have a packet which has been written that it's active. So that type of yeast is alive and you purchase it as it is. So yeast is killed during cooking but adds in all the yeasty flavor to bread. Yes, of course. Whenever you bake a bread, what do is you allow that yeast to grow, right? You give sugar, you give other nutrients to it. We keep that for 1 hour or 2 hours, right? It rises up, it leaves up, and what happens is after that, when we bake it at high temperature for cooking, like when we cook it, what happens is due to the temperature, the yeast has been killed, right? But yeast has done its work. It has risen the bread. It has made the bread more softer. It has given its flavor or taste to the bread, right? So this is the work that baker's yeast does to the breads. Then comes brewer's yeast. In brewer's yeast, as the picture says, okay, you may have seen such different types of bubbles like structures or such crowded microorganisms, right? In a wine bottle, beer bottle, or even if you have visited any wineries, you may have seen such type of image or things you can see over there. So this type of brewer's yeast can be purchased alive only, but it is used to brew beer. Okay, so the dead yeast cells left over from the brewer's process can be consumed and as a nutritional supplement but have a very bitter taste. Now bitter taste, you can imagine why beer or wine has some amount of bitter taste, right? So it has some fruity taste also because of the fruits which we are using for making that wine, but still it has a bitter taste, and that is because the yeast. Because yeast when grows leaves a flavor behind in its liquid, right? That is because it is consuming the sugar and converting it into the alcohol. So this is the work of brewer's yeast, which is used to bake to brew a beer, right? Then nutritional yeast, grown specifically to be used as a food product. So as I said, nutritional yeast is a different type of yeast which is generally used as a food product, or you can even say that it is generally being added in every food product because it's an edible part. Okay, you can directly consume it with some amount of vegetables or other material which we eat in our day-to-day life. So it is generally used in cooking and has a very cheesy, nutty, and a flavory flavor, which is much different from the brewers and the bakers yeast. Now, this was the introduction of yeast, like where the yeast has been used, the products which we can make, or the products which are actually being consumed by us, which is produced by the yeast. Now we are going to study the protein content of this yeast. So in this we are going to study the protein content of yeast, and we are going to understand some nutritional facts of the yeast, like how it is better from the other protein content which we are actually eating nowadays. Okay. Now, to be very specific, we have studied different types of yeast, right? But we are going to go further with a specific type of yeast. So I have chosen baker's yeast because it's the most common type of yeast which we generally know and we use, and yes, we consume bread, so we are consuming this type of yeast, right? So baker's yeast is a great source of protein, B vitamins, and trace minerals. You may have known that yeast is a single cell protein, right? So it produces protein, or it has protein, but it is also a great source of B vitamins and trace minerals. So nutritional yeast contains more B vitamins than the unfortified varieties. Means what? Like we are using baker's yeast, right? But we are not using nutritional yeast. But still we should know that why nutritional yeast is directly being consumed as a food product, right? So nutritional yeast as compared to the baker's yeast has more content of B vitamin, and that's why it has been consumed as a food product in many countries. So it has some amount of extra vitamins, or you can say extra protein content. Okay, and still the other varieties still contain a moderate amount of B vitamin, which form naturally as a yeast grows. So naturally, when a yeast is grown, to let it be baker's yeast, brewer's yeast, or nutritional yeast. So what happens is naturally there are some B vitamins, proteins which are in grown, right? So as compared to that, nutritional yeast is more beneficial and contains more amount of yeast. Now, it it has protein, B vitamins, and trace minerals. Let's see how much amount of this all nutrition does it have. So first is it contains complete protein, right? Baker's yeast contain all 9 essential amino acids. We are going to see what 9 essential amino acids it actually contains. But it has all essential 9 amino acids which one should actually consume in its day-to-day life. Okay? So 1 tablespoon of the yeast, baker's yeast, contain 2 grams of protein. So you may just imagine that what amount of yeast you are using when you are baking a bread. Okay, as per that you can calculate actually how much amount of protein you are consuming by using this baker's yeast. It contains many B vitamins. So 1 tablespoon of baker's yeast contain 30 to 180% of B vitamins. Like if there is 100 grams of baker's yeast packet, okay, so amongst them 30% of that 100 grams will be B vitamin, and it is essentially rich in thiamine, riboflavin, vitamin B6 and vitamin B2. And last is the trace minerals. So 1 tablespoon contains 2 to 30% of trace minerals such as zinc, manganese and molybdenum. Okay. So for trace minerals are involved in gene regulation, metabolism, growth and immunity. So this Becker's is going to provide you some amount of immunity also. It is going to give you more good content of vitamins also and proteins also. So it's very necessary that you should consume it, and it is also important that how much amount of Becker's you are consuming. Now, as you can see, there are fats, saturated fats, cholesterol, sodium. These all things are actually nutrition facts. Like whenever you buy a product or whenever you buy a baker's yeast packet, okay, in that packet or on that labeling, you can see such tables, right? You may have seen, or if you have not seen, you should see this next time whenever you buy that. So suppose it's a 100 gram of packet, okay? So you can see this 100 gram of packet is serving you 107 amount of calories, okay? And protein is 8 grams. You can see in the label. So in the right hand side image, you can see that there are proteins which is 8.4 grams, fats which is 1.9 grams, carbohydrates which is 18.1 gram, water of course, and other minerals, or you can say B vitamins like just we have seen. So this amount has major impact on your baker's yeast for growing, for keeping it active and for consumption also. Okay. We are going to focus on the protein completeness. So protein completeness means what? A protein is called complete when proportional to all overall amino acid content, right? It has enough of yeast essential amino acids, and of course we have discussed that there are 9 essential amino acids which are present in this yeast, right? Or protein content which is present in the yeast, it has 9 essential amino acids. So Baker's yeast has a complete source of protein. Okay. So this essential amino acids and this all contained in the yeast makes that yeast a complete source of protein. As you can see in the table, you have leucine, you have valine, histidine, lysine, isoleucine, tryptophan, threonine, methionine, and phenylalanine. Right? So these 9 amino acids are present, and you can even see the amount in mg per gram of proteins which is present in that baker's yeast. Okay. Now in this table we can see, or you can see that we have compared the baker's yeast with some amount of foods, like you can see cheese, broccoli, rice, oranges, right? So we have compared the content of protein with the content of protein in this food also, so that you can know that how our protein, or you can say yeast, is comparatively higher than those food protein content. Okay. So the protein content in baker's yeast is 20%, whereas which is 3 times more than a broccoli. Okay, we said that broccoli is very good in protein, or it's more good in nutrition. But if you can see over here in the table, protein content in yeast baker's is 3 times more of that broccoli. Okay, same is with fats, carbohydrates, calories, sugar. But here we are focusing proteins in this practical. So I'm going to explain only about the proteins. Okay, you can refer this chart and know more about the baker's yeast food container. Now, here are practical stuff. We have studied what yeast is, we have studied what are the protein contained in the yeast, right? We have studied baker's yeast. Now we have to take that baker's yeast and we have to perform biuret test, which will give you exactly what amount of protein content is present in your yeast. Okay. So in this practical, we are going to study what is biuret test and principle of biuret test and how it works. And of course, we are going to estimate, we are going to know the method by which you can estimate the protein content in yeast for this practical, and this method can be used for estimating of protein content in any of the material. Okay. Biotech test. So the aim of our practical is what? To estimate the protein content in the yeast, which is a single cell protein, by biotech method. Okay. So let's focus to the principle of this method. This method is a general protein assay. Okay. There are many other methods, like there is Folin-Doyle method, there is Bradford method. Okay. All methods are used for protein assay, or you can say estimate some amount of protein in the biological sample or any other sample you want. Okay, so here we are going to focus on biuret method. Peptides containing 3 or more amino acids. You all know what peptides is, right? Amino acids bind with each other, or they are in a chain, right? So that chain is called as peptide chain. So they are bind to each other, and that residue, that peptide chain, forms a colored chelate complex with cupric ions. Now, copper ions. You may have known it has a blue color, right? So when your peptide chains gets attached, and you can say get bound with that copper ions, it forms an alkaline environment. Okay, which contains the sodium, potassium, tartar in solution. And this is what a biuret reaction is. The biuret reaction contains what? Your amino acid chain, okay, which is bind to an copper ions, and it forms a color because of sodium, potassium, tartar. Okay, this is because it is chemically similar to a complex that forms with the organic compound biuret and the cupric ions. We are going to see how it interacts with copper ions in this slide. So biuret, a product of excess urea and heat, and it reacts with copper to form a light blue tetradentate complex. As I said, copper ions, right? Copper has blue color. So it is going to have a blue color. So biuret reagent is going to have a blue color, right? Plus, when your peptide chain, or you can say your protein content in any of these sample is going to come and bind with the copper ions, it is going to change the color. So it is going to change the color to dark blue. Now you can see how a biuret reaction actually takes place. And here you can see 2 amino acids. Okay, there is written 2, and there are 2 amino acids bind to each other. We are adding copper ions. Okay, and in the right hand side picture you can see, or in the right hand side reaction, you can see there is a violet color complex which is formed. Right? What is this violet color complex? So, as I said, your copper ions or your biuret reagent is of blue color, right? So, that blue color is converted to the violet color when your amino acid or protein content in the sample comes and binds to the copper. Okay, this reaction is formed, and because of that, it converts the color to violet. Now, let's see the procedure. I have added a table which will make you more simpler, which will make you understand more simpler that how we are going to perform this practical. You have to take 5 tubes. First tube will be blank. Second, third, and fourth will be standard samples. Okay, like standard protein is present, right? So we are going to vary the samples, vary the concentration of that protein, standard protein, and the last fifth tube you will have a test sample. So what is your test sample? Your test sample is yeast, right? So likewise. So your first tube is blank. In that blank, you are not going to add anything. You are not going to add standard protein. You are not going to add yeast samples. Why? Because that is blank, right? So we have to analyze water. You can add distilled water and directly the reagent in it. Okay. So there are four chemicals. So you can say there are four solutions which you have to add in your tubes. First is the concentration of BAC. So BAC is a protein, right? Which is a standard protein which is generally used for your practicals of estimating protein content in any of these samples. So here you can see we have varied the concentration of that standard protein. So here you can see in first, second and third. Okay, you have numbered the tube blank 1, 2, 3 and 4. So in first, second and third tube you have different concentration of BAC. So in first tube you have 2 mg per ml concentration of BAC. In second tube you have 4 mg per ml concentration. In third tube you have 8 mg per ml concentration, right? So we have doubled the concentration in each tube. Okay, and then you have to actually add the stock. Stock of what? Stock of BAC. You have varied the concentration. You have labeled the tube as per the concentration of standard protein. Then you have to add the stock of BAC. We have to make the stock because we can't directly measure 2 mg, right, per ml of BAC and add. So we have to make some mg stock earlier, and then we have to add that much ml of stock so that it will become that much mg, or it will match the concentration of that BAC. Now amount of stock you have added is, in first tube you have added 40 mol, okay, which comes to 20 ml, 2 mg per ml. Then 80 in second tube you have to add 80 mol. Now you can see that we have doubled the amount of stock, right? That means if we have 40 mol means 2 mg per ml concentration. So you have to double the stock, means 80 mol means 4 mg per ml concentration, right? So whereas we have also doubled the next third tube concentration, whereas we have added 160 mol of stock solution, which will give you 8 mg per ml concentration. Okay. Now here you have added the standard protein in first, second and third tube. Now you have to add in fourth tube. You have to add your standard. You have to add your sample, right, which is your yeast. So you have to add 200 ml of sample solution in fourth tube. Now why 200? Because we have to volume up our tube. Okay, volume up our samples with 200. So we have directly added 200 ml of yeast samples in the fourth tube because we don't know what is the concentration of that protein in that yeast, right? Because of that. So now you have to volume up all the tubes till 200 ml, right? So in black you have not added anything yet. For that you have to add 200 ml of distilled water. And now in first tube you have 40 ml of stock solution or BAC solution, right? So to volume up it till 200, you have to add 160 ml of distilled water. As per that, to make the volume 200 ml, you have to add the distilled water as per the need. Okay. Then after that, after you have volume up all the tubes till 200 ml, you have to add the biotech reagent which we have made earlier. Okay. So for that, what will you need is, you need copper ions, right, and sodium potassium hydrogen. So it will give you a blue color complex, which so the reagent, biotech reagent is of blue color. Okay, and then you have to add 2 ml of biotech reagent in each tube. After that, you have to incubate your tubes for 10 minutes at room temperature. Okay. So once you have incubated it at room temperature, you have to observe the color first, and then you have to go for analyzing the observance of that samples. Okay. So let it be very clear that in this practical we are going to study both qualitative and quantitative analysis of the product. Qualitative analysis means what? You are going to observe the color change, right? So if your protein sample, or you can say your test sample contains protein, then the color will be converted, the reagent's color will be converted to violet. But if your sample doesn't contain any amount of protein, then what will happen is it will not change the color and it will remain blue, right? So this is how you can do the qualitative analysis, and quantitative analysis means you have to know the amount of protein present in that test sample. So for that you have to calculate the observance, or you have to calculate the optimum density at 540 nanometer. Okay. Now in this image you can see that here we have a standard line, right? So here we have taken the observance of protein solution at 540 nanometer, and we have got a straight line. As in earlier slide we have seen, we have varied the concentration of standard protein, right? 2 mg per ml, 4 mg per ml and 8 mg per ml. So as per that, what will happen is you have to plot a graph. So that graph will be as in x-axis you will have your protein concentration, standard protein concentration, and in y-axis you will have the observance, right, which you have taken at 540 nanometer using a spectrophotometer, or you can also use a colorimeter. Okay. So if you have taken that observance, you have to plot that graph and you will get a straight line. What you have to do is you have to plot these standard concentrations, okay, and you will get a straight line. As you can here see the violet line which is going straight. So you have taken 2, 4 and 8, right? Here they have taken 2, 4, 6, 8, 10. You can vary as per the concentration you want, but we have just taken 2, 4 and 8. So you have to plot the graph and you will get a straight line. And now your test samples, right? So in the earlier table, you can see that we have taken test samples, right? And now we have also taken the observance of the test samples at 540 nanometer. So now what you have to do is whatever the observance you have got, you have to extrapolate it on this graph. Okay. So suppose you have got 0.354 OD of that test samples. So at y-axis, wherever the 0.354 comes, you have to extrapolate it and know the concentration of your protein or test samples. So now here you can see in this graph they have extrapolated the OD, right, of of test samples, okay, and the circle on the x-axis you can see over there, that is 4 mg per 200 ml. Okay, because why 200 ml? Because we have added the test samples 200 ml, right? So the mg which you will get, or concentration which you will get, will be mg 4 mg per 200 ml. Okay, so this is how you get a protein concentration by extrapolating your OD on the standard graph. Okay, now you have to know the protein concentration, actual protein concentration of your test sample. So for that, what you have to do is there is a formula. Okay, you have to calculate it using a particular formula. That is a concentration of unknown in mg. Now you have known the concentration of unknown, right? That is 4 mg per 200. So 4 mg you have to write it over there, divided by volume of samples in ml, that is 200 ml, right? You have taken 200 ml of your sample into 5. Okay, so whatever will be the answer by calculating this, you will write it as a protein concentration of test sample is equals to that much amount of mg per ml, right? So this is how you can calculate the concentration of protein of your sample. And this is the observation of your experiment. Like here you can see the qualitative analysis they have done. Qualitative analysis means what? You have to just observe the color change, right? So in first tube, second tube, third tube. In the table we have studied before, in that table in first tube you have 2 mg per ml of standard samples, in second you have 4 mg per ml of standard samples, in third you have 8 mg per ml of standard samples, right? So as per the concentration increases, of course the color will change, right? So in first tube there was least amount of protein concentration. So you can see the color has not changed, it's blue. And in second you have fold, you have doubled the concentration of protein. So you can see the blue color has been little bit darker, right? And in third, you have the maximum amount of protein concentration and it has been converted or it has been turned to completely white. So this is how you can observe whether actually you have protein in that samples or not. Okay. So how will you interpret these results? The biorate method is carried out by performing a set of solutions with protein concentration and mixing them with the biorate reagent. And the standard curve can be made and the concentration of Baker's yeast protein sample can be derived from the standard. Okay, so this is how you can estimate the protein concentration of your sample. Here we are talking about the sample that is yeast, right? So yeast is very simple to analyze. Even any sample, if you want to analyze the protein content by biorate method, you can of course analyze. Okay. Summary of this practical is, first of all, you have studied the yeast as a single cell protein its reproduction system, that is studying how a two cell or it divides in which form that we have studied. Then protein content in the Baker's yeast, right? It has protein, it has B vitamins, it has trace minerals, and how much amount of protein it has, that also we have studied. And at last, the protein estimation of yeast by biorate test. Okay, so these were the main agendas of our today's practical. Thank you.